Now, there are three types of the small mutations. So basically you have substitution, that's the first one. You also have insertion or a deletion. So you know that either insertion or deletion can shift the reading frame of your original gene. And we have mentioned before that you have, for example, here was an insertion of an extra base. Now your code does not read for tyrosine, it will read now for leucine because through their ribosomes. It does not know that this base wasn't there to begin with. It will just read, 3 bases at a time. The large unit of the ribosome and the small unit of the ribosome, when it's translating the messenger RNA, once it's translating the messenger RNA, it doesn't know that a particular base shouldn't be there. So it will just read 3 bases at a time and translate what is on that sequence. Okay, so these are some phenotypes of genetic mutations that you can have. So you have flower mutation that you transfer from a wild type to a mutant type. You have a reverse mutation that you transfer also the mutant type to a wild type. You have a missense mutation. Now, this one you have to know, the missense mutation. That will do what? Right, will change the codon and it will code for a different amino acid. That's a missense mutation. Now the nonsense mutation, what does it do? Makes a stop codon. And then you have a silence mutation, silent mutation. What does that one do? Doesn't affect the codon for the final amino acid. It skips the same amino acid, but the gene got mutated. And then you have the neutral mutation that is close to nonsense mutation, is related to the nonsense mutation. So what is a neutral mutation? Similar amino acid. You keep the same function of the protein, but you have a different amino acid in a specific place where the gene was mutated. So those would happen when you change from one amino acid that has similar chemical properties as another amino acid. If you have a basic amino acid, you have another basic amino acid inserted in there. Oh, is the both of them? Yeah. Because if it's only one, then that can be easily repaired. Okay. Because they have the polymerases. They are usually patrolling for changes or problems with the DNA after replication or this type of mutation. As I'm going to say today with the DNA repair mechanism. But if it happens on both sides, then you don't know what to repair. You don't know there was a problem in the first place. Okay. So that's when you have a problem. Now here are other forms of mutations in the gene. Now this is a small gene mutation because it happens with a very small segment of a particular gene, but is also a large gene chromosomal mutation because you can see this in the microscope. So this is a case of Fragile X syndrome. And what happens here is that when you have the replication of the chromosome, the replication of the genes, we don't really know why that happens, but a particular segment of a gene that has repeat sequences, they get a lot more repeat sequences that they're supposed to have in the first place. And depending on the number of the repeat sequences, some numbers of repeats are normal. Like, for example, everybody would have, on this particular gene, everybody would have about 10 copies of this trinucleotide repeat, CAG, on chromosome 8. So if you look at everybody's chromosome 8, you'll find about 10 copies. Sometimes you find 8, sometimes you find 20 or so. But if you find over 100 copies or 200 copies of this particular segment of this particular trinucleotide, then you're going to have a problem, because this change in the protein sequence, change in the protein structure, is going to cause malfunction of the protein and is going to lead to deleterious phenotypes. Now, this trinucleotide repeat expansion, or the Fragile X, is one of the most common causes of mental retardation. One theory of why this happens, why this increases, nobody really knows. Now, one theory of why this happens is a DNA polymerase slip during replication. Okay, so think about it. Now the cell is ready to divide. So there goes polymerase and it's replicating the DNA. Now, how do you have a picture in your head? Just imagine that you're working out on the treadmill and every step you take is corresponding to one turn of the belt on the treadmill. Now, sometimes it happens that the treadmill is not regularly adjusted and the belt will just slip out of place. So then you have to catch up with it a little bit and eventually you start replicating again or running again. Now, this could happen with the polymerase on the DNA while it is replicating. Sometimes, especially on those repeat sequences, it will pass a little bit faster or cause some type of hairpin formation. Remember, those are repeat sequences, and the C and the G can lead to a hairpin formation. So that will be the slipping on the belt, and then it will catch up again with the rest of the repeats, and it's going to replicate again. And this somehow, this change, this loop is not detected for correction. So this is one of the hypotheses of why this happens. Now, you can also imagine that if a person is prone to that kind of error, you know, if the person, the DNA polymerase in this person is prone to this type of error, so chances are that this type of error is going to happen more and more often as the generations come along. So this causes that anticipation, the symptoms of anticipation. And what that means is that if the grandfather had the disease, had the syndrome, the father is going to start showing symptoms of the syndrome at an early age. So here are some of the syndromes that are caused to trinucleotide repeats. You have muscular dystrophy, spinal bulbar muscular dystrophy, Fragile X is right there, the most common. You also have here the Huntington disease that I just mentioned, and several others. These are just the most common. Here, epilepsy. One type of epilepsy is also caused by a nucleotide repeat. It's not a trinucleotide, but it's a nucleotide repeat. Now, how is it? Remember I mentioned that if you have a DNA mutation, the mutation is permanent. Because if it is a mutation, it was not detected by the repair mechanism. So it got mutated, it's permanent. Now, how is it that it becomes permanent? This is a common theme that you're going to see over and over. A change occurs on one nucleotide, and then replication happens. Now, if you have here a change in one nucleotide, and the replication is happening, so the next time that cell divides, is going to use this mutation as the template DNA. So now the cell doesn't know that this was a mutation, so it's going to just replicate whatever is in there. So you don't know that one side here that replicated had the non-mutant DNA, and then the other one has the permanent mutation in there. Again, causes, then remember I said you have types of mutations that can be induced by the environment, including drugs and radiation, everything else. And you also have spontaneous mutation that causes errors in the DNA. Now, what are the types of spontaneous mutation that can cause that type of DNA damage or mutation? You can have error in the proofreading of DNA polymerase, which means that, remember, some of the polymerases have the proofreading 3 to 5 prime and 5 to 3 prime error-proof proofreading. If you have errors in the proofreading, that can introduce a mutation. Also, if you had the strand slippage, as I just mentioned with the treadmill, that will introduce mutations. So here, as an example, if you have defective proofreading, and you have here an insertion of a base, and this insertion of the base did not get corrected, so it is going to keep the mutation, just like the change in the base after cell division. And then keep in mind that if that happens in the gamete cells, so the offspring is going to have the mutant genes. And also, it's important to keep in mind that mutation is natural. As I mentioned, it's the only way that you can have evolution. And usually you have a lot of mutations during the day, and you have, as I mentioned in the lab, you have an average of 50 to 70 mutated bases when you compare your chromosomes to your parents' chromosomes. So in talking practical terms, we're all mutants. We all mutated our DNA from our parents. And we have kids, they're all going to be mutants. So you have other forms also of spontaneous mutations. So there is one form that you have to keep in mind. It's a tautomeric shifts. Now, what does that mean? Do you all take organic chemistry? Most of you, no? Yes? Okay. The tautomeric shifts, the tautomer is a form of a molecule that can be in equilibrium with a more stable or more encountered form. So what happens when it shifts to the rare form? Well, it can base pair with something that is not supposed to. So you can imagine that this form of mutation is should be fairly common if you have this equilibrium situation in the body. But it so happens that this is very rare. Now, imagine if this happens all the time and you always have a lot of bases that are in the tautomer form and then you mutate all the time. That is not a very good rate of evolution. So you can imagine the body has ways of, the cell has ways of holding down this process. Now, do you know who hypothesized about this type of tautomeric shift and DNA mutation? Yes, who else? Watson and Crick.